Good afternoon, everyone, and thank you for joining us for today's webinar. Today's webinar is the 10th webinar in our opioid webinar crisis series. Rates of opioid use and misuse have reached epidemic proportions and are impacting many aspects of both criminal justice and forensic science programs. Opioid addiction is the driving force behind this increase in use, with nearly 3 million Americans reporting a substance use disorder to prescription pain relievers or heroin in 2015, fueling a steady increase in fatalities to an estimated 91 U.S. deaths daily. These rates are not slowing, rather they are doubling, quadrupling, or undergoing even more alarming increases in some areas. Law enforcement, medical professionals, laboratories, and legal agencies are battling with unmanageable caseloads, economic shortfalls, and challenges for safety, analytical preparedness, and basic education and training. Reliable surveillance and intelligence are needed more than they have ever been, confronted with the fast-paced emerging drug life cycles. The legislative quagmire is just as burdensome, as policy change cannot happen without the data to support the change. This webinar series will bring a multifaceted perspective to how diverse criminal justice disciplines are addressing these challenges, sharing their knowledge, and advancing science, technology, and law. Dealing with the impacts of the opioid crisis to the criminal justice system requires better reporting, surveillance, research, technology, and policy than are currently in use. The need to understand the epidemic and its effects goes beyond knowing your own profession. It takes a global perspective to fully act and make a difference. My name is Josh Vickers. I will be the moderator and technical host for our webinar today. Today's webinar is the industry's role in responding to the opioid crisis. We have two wonderful speakers from Cayman Chemical today. Our first speaker will be Donna Ayula. Donna is the Director of Forensic Chemistry at Cayman Chemical. She earned her PhD in Synthetic Organic Chemistry from the State University of New York at Stony Brook and did her postdoctoral training at the University of Miami. Dr. Ayula then worked for 7 years in the pharmaceutical chemistry as an R&D scientist and medicinal chemist prior to joining Cayman Chemical in 2007. In addition to her expertise in emerging drugs and abuse, drugs of abuse has been sought by several news media outlets, and she has shared this knowledge by providing training to state forensic crime labs. Our second speaker today will be Roxanne Frankowski. Roxanne is currently the ISO Quality Manager at Cayman Chemical. She earned a Bachelor of Science degree in Chemistry from Siena Heights College, and then her Master's of Science degree in Forensic Science from Michigan State University. For 12 years, Ms. Frankowski was employed as a forensic chemist with both the Michigan State Police and Drug Enforcement Administration, where she was responsible for the authentication, distribution, and inventory of the DEA's reference material program prior to joining Cayman Chemical in 2015. Throughout her career, Ms. Frankowski has had the opportunity to present on a variety of topics, including the characterization of reference materials, and has also provided training to state, federal, and international forensic chemists. Now, before we do get started, the FTCOE's disclaimer, the opinions, findings, and conclusions or recommendations expressed in this presentation are those of the presenter and do not necessarily reflect those of the Department of Justice. Any products and manufacturers discussed are presented for informational purposes only and do not constitute product approval or endorsement by the U.S. Department of Justice. And now I would like to welcome everyone to our presenters today. Our first presenter again will be Dr. Donna Ayula, and then after that, Ms. Roxanne Frankowski will finish us out. Donna, if you'd like to go ahead and get started.
Thank you for the introduction, Josh. Can you hear me okay?
Yes, ma'am. Your audio sounds great.
Okay. Well, thank you for the introduction, and thank you to everyone at the Forensic Technology Center of Excellence, the NIJ, and RTI for the invitation to participate in this extremely important webinar series. Roxanne and I are very happy to be able to share with you industry's role in responding to the opioid crisis. Before I begin, since I am aware that we have a very diverse audience, I'd like to provide a little bit of background on the company curiously called Cayman Chemical. Cayman Chemical is headquartered in Ann Arbor, Michigan, and we were founded in 1980 by Dr. Kirk Maxey. Since then, our mission has been to help make research possible by providing the research tools for advancing research in a wide variety of disciplines. Our expertise in endocannabinoid synthesis led our customers to begin to request other types of cannabinoids, such as synthetic cannabinoids that were emerging on the recreational drug scene well over 5 years ago now. So the increased demand for reference standards of other emerging designer drugs, including cathinones, amphetamines, led Cayman to create a dedicated forensic chemistry team, as you see pictured here. They're an amazing bunch, and I am honored to work with them every day. We have grown over the years to be an internationally known and leading manufacturer of highly pure analytical reference standards. We are a team of diverse backgrounds, including synthetic organic chemistry, analytical chemistry, medicinal chemistry, and forensic chemistry. In addition to providing reference standards, we offer free web tools, which Roxanne will review later. We are proud of the value we bring to the forensic and toxicology community, and we are very proud of our customer service. We are also ANAB accredited for ISO 17025 and ISO Guide 34. Our customers include state forensic crime labs, federal agencies and border patrol, government and private toxicology labs, international government agencies, instrumentation companies, device manufacturers, as well as academic research institutions. I'd like to point out that all of our customers are put through a very thorough vetting process. So while we sell small amounts of analytical reference standards to law enforcement and accredited research institutions, a Google search for research chemicals for sale pulls up dozens and dozens of not so legitimate quote-unquote gray area websites. The ease with which novel psychoactive substances can be sold and purchased to anyone on the dark web, or even openly as you see here, has certainly fueled the current illicit drug trafficking crisis that we see today. A Google search for carfentanil for sale pulls up many vendors selling bulk quantities. If you look at this site here on the upper left, for example, you see that they have an option to purchase 3 kilograms of this deadly opioid. The site on the right has even reviews. You may not be able to read the text, but this guy who calls himself Steve Opiate, probably not his real name, just in August of this year wrote, quote, "Everything went smoothly and as expected. Shipped same day as order was placed on Monday, received Thursday to Northwestern USA. Have only taken one tiny hit, and I was greeted with a blessing of extreme euphoria and the welcomed warmth of an opiate security blanket. Would recommend vendor and may use again," etc. So this gives you a little taste of what law enforcement is up against. As you might be asking yourself after seeing all of these vendors openly selling powerful drugs, where does the clandestine chemist get the knowledge of which opioids to make and how to make them? Well, a Google search for fentanyl synthesis, for example, gives you your answer. The internet is a wealth of information. In mere seconds, anyone can pull up the synthetic routes to prepare these substances. Included in this screenshot is the a U.S. patent that lists fentanyl analogs that were filed in the 1980s by pharmaceutical companies conducting legitimate research in their pursuit of safe and effective analgesics. So the list of opioids to make and precisely how to make them is out there in the scientific literature, and there's not much we can do about that. So with all these challenges, what role does industry play in providing the tools the forensic community needs to combat this epidemic? Industry is on the front line in that combat. The speed with which new analogs are appearing their increasing potencies and unknown toxicities has forced us to be more innovative and more collaborative. For example, there's increased demands on industry for novel instrumental techniques and instrumentation. There's an urgent need for more and better presumptive field test kits and devices capable of detecting the newest psychoactive substances. And as illicit drugs become more potent and more mixtures are encountered, there is the need for enhanced limits of detection. Drug screens need to have the most up-to-date and relevant metabolites, and spectral libraries need to be current. And lastly, analytical reference standards need to be prepared more rapidly. So as a reference material manufacturer, Cayman Chemical has had quite the challenge to meet the forensic chemistry and forensic toxicology demands for the latest opioid substances on the street and their metabolites. While there are several synthetic opioid chemical classes, the fentanyls, as you see here, have been the most abundant, and so I would like to discuss them a little bit further. By now, I'm sure everyone in the audience is familiar with the chemical structure of fentanyl and the various regions where that backbone can be modified. Let me pull this up. So there is what is referred to as the acyl group or the amide. For example, there are many fentanyl analogs where there is something other than a C2H5 here. Most, if not all, of the illicitly prepared fentanyls have a piperidine ring that may or may not have a substituent. There are two arrow rings on the skeleton. One is part of what we refer to as the aniline ring. This is where the phenyl ring is directly attached to the amide nitrogen. The other phenyl ring is part of the N-alkyl moiety, usually spaced from the piperidine by 2 carbons. So one of the reasons there are so many fentanyls being trafficked is because they are relatively easy to make and easy to synthesize if you have access to the synthetic precursors or the building blocks to make them. So here is a scheme to make fentanyl that you can find on the internet. This particular route is a 3-step process. The first step is an alkylation reaction where commercially available piperidinone is treated with phenethyl bromide. And this reaction provides an intermediate that is abbreviated NPP. The next step is a reaction called a reductive amination reaction. And here the ketone NPP is treated with an aniline in the presence of a reducing agent to give a compound called 4-ANPP. 4-ANPP is both a precursor to fentanyl and its many analogs, but it's also a minor metabolite of those fentanyls. And the final amide formation can be accomplished by using the appropriate carboxylic acid, anhydride, or acid chloride. So one thing I like to point out to the general audience like we have today is that while some people refer to the tweaked or modified fentanyls as fentanyl derivatives, you cannot simply convert fentanyl itself to any one of those analogs. Each one must go through its own unique preparation. For example, any fentanyl analog that has a substituent on the piperidine ring here, such as 3-methylfentanyl, requires a starting piperidinone possessing that specific substituent. Any derivative, for example, where there are substituents on this arrow ring necessitate the use of the corresponding alkyl halide in the first step of this particular synthetic route. There are many commercially available aniline derivatives, and their use in this second step determines which aniline-modified fentanyl analog you see here. The choice of acid chloride, carboxylic acid, or anhydride in this final step determines which amide group you see here. And if you look on my next slide, there's countless acyl modifications that can be carried out. If a clandestine chemist were to purchase or make that 4-ANPP intermediate, there are almost no limits to how this fentanyl skeleton can be tweaked in an attempt to avoid detection and prosecution. So my take-home message by going over all this organic chemistry is that the building blocks are extremely varied and expect more fentanyls in the future. So clearly the emergence of so many designer drugs is a challenge to the forensic and toxicology community. There are not just the fentanyls to keep on top of, but there are the U47700 analogs on the street. There are other opioid chemical classes, and we can't lose sight of the novel amphetamines, cathinones, tryptamines, synthetic cannabinoids, etc. as well. So how has this impacted Cayman Chemical? We have had to keep up with the rapid pace with which these analogs appear. We have had to develop a consistent and clear way to name these analogs, which I'll get to soon. As time and resources are precious, we've had to prioritize which standard to make. Particularly the CRMs, which we will describe soon, are more lengthy to produce. We've also been asked to make metabolites for the toxicology community, often with little to no published metabolism studies to go by. We have had to develop protocols to manufacture these compounds safely. Our regulatory department is busier than ever. We have had to update our GC mass spec library more frequently, and we have actually had to aid instrumentation companies in building their own libraries. So our customers go to our website and they purchase the standards they need to complete their casework or conduct their research, but often they don't think about what goes into having those standards available. So I created a simple flowchart to give you some insight. Imagine a long queue of items that we wish to offer. So compound A, I'm losing my arrow, here it is. So imagine a list of compound A, compound B, compound C, metabolite 1, metabolite 2, compound A, D3, etc. If you take any one of them, say compound B, we first have to start by designing a synthetic route to make that molecule. The synthesis is then initiated, and sometimes significant research and development needs to go into it. Along the route, especially for the final target compound, there are lots of purifications and characterizations to be sure you have what you set out to actually make. Once the synthesis lab is done, one of our quality control departments runs a battery of tests, and Roxanne will describe this in more detail a bit more. If everything passes, a certificate of analysis is generated and the standard is available on our website. If a compound is slated for CRM production, additional tests are necessary prior to and after making that solution before that CRM can become available. So the take-home message is that for any single compound, this process can take anywhere from weeks to months. I know the title of this slide has been a curiosity for many of our colleagues out there. So how do we keep up with the market? And how do we prioritize what we offer on our website? Well, the answer is not a secret. We collaborate with forensic chemists and toxicologists throughout the world who tell us what they need. We attend forensic conferences, we read published literature, we also monitor drug forums, chat rooms, and foreign websites selling drugs to see what might be heading our way next. The role of industry is to be a close collaborator to the forensic community during the opioid crisis. I can't stress that enough. It's only when the strengths of all parties, government, academia, industry, etc., come together that we can put forth our best defense. I now like to provide 3 quick case studies that I think illustrate the positive and collaborative relationship Cayman has had with the forensic community. U47700 is a synthetic opioid developed in the 1970s, but never advanced to clinical trials. It has been implicated in many overdose deaths in the past few years. Back in October of 2015, I was presenting at a regional forensics meeting, the Southern Association of Forensic Scientists, and Joshua Yohanan asked me about a compound he called U47700. Hope you're out there, Josh. Hi. I'm not sure exactly where he heard about it, but it certainly wasn't on our radar. I jotted down the name and I took it back to Ann Arbor with me and added it to our long queue of compounds. But it was only after several additional inquiries for this molecule, which coincidentally is isobaric with another known synthetic opioid, AH7921, did we actually set out to make that reference standard. The point of bringing up that story is to illustrate both how we decided which standards to make and how important and influential our forensic colleagues are. So this leads me to case study 2, carfentanil. This example further illustrates the critical role that private industry plays and the need for rapid communication. It was probably late spring, early summer of 2016 when we received our first inquiry about carfentanil. I'm sure someone, probably me, looked at what was reported to be the potency of this compound and just said, sorry, we don't offer that standard, and we went on with our work. But then we had 5 calls and emails, then 10, and the forensic community grew more desperate and frantic for help, and we knew we had to act. So we had multiple meetings that came in that centered on, how do we do this? Do we even want to do this, knowing the potential personal safety risk? But after careful planning with our safety team and some very nervous days back in October of 2016, a team of very brave chemists, including Roxanne and myself, and I have to give a shout out to our pilot, Chung. We ran the final step of the synthesis. We purified the compound, characterized it, and we QC'd it. The standard was made available, and those who were in desperate need of it were able to purchase it. So I'm really proud of our team for stepping up and providing that. So last case study, last year came and received an email from a toxicologist in Alabama. And she had several death cases tied to a substance that she could not identify in any spectral library. She reached out to us for our expertise, and she shared her data. One of our scientists looked at that data and proposed that her unknown was a fluorinated acryl fentanyl analog. So we quickly synthesized that compound, and we shipped it out to her for confirmation, but it wasn't a match. So in January of this year, we reopened that investigation. She now had even more death cases piling up, and we were ultimately able to decipher some minor GC-MS spec fragments to discover that her unknown was methoxyacetylfentanyl, as you see drawn here. And since then, methoxyacetylfentanyl has been identified in many additional states. So by sharing these 3 examples of how private industry came to the aid of the forensic community, worked closely and successfully together, I hope that you recognize that we are listening. My advice for strengthening this partnership is to reach out early and reach out often. Let us know what you need. We are listening and we will respond. What you are seeing in Alabama, Oregon, or Pennsylvania, for example, will most likely show up elsewhere. So the sooner you make us aware of what you need, the sooner we can get working on those reference standards. So before I pass the microphone on to Roxanne, I would like to discuss the naming convention for fentanyl analogs that we have adopted. So as mentioned earlier, there are several regions on the fentanyl skeleton or backbone where modifications occur. This poses a challenge, for example, when one talks about 2-methylfentanyl, let's just say. There are 5 theoretical possibilities for where that 2-methyl group could be. So we had to come up with a way to name these emerging fentanyls so that we were all on the same page. So we often see examples of fentanyls where that C2H5 ethyl group that you would normally see here off the amide carbonyl is replaced with a different alkyl chain or heterocycle. So these are easily named by placing the known chemical term for that moiety ahead of the word fentanyl. So here are some examples. So if you were to replace that C2H5 ethyl group of fentanyl with a cyclopropane ring, sorry, my screen just went blank, there we go, you get the name cyclopropylfentanyl. If you replace that C2H5 ethyl group with, let's say, just a methyl group, then you get what we call acetylfentanyl. If you replace the ethyl group with, let's say, a phenyl ring, you get phenylfentanyl, and so on. So what about substituents? And so how are compounds like on this slide named where we might have then more than one change, let's say, to the fentanyl backbone? So here is the convention that we have adapted here at Cayman to make it easier for the forensic community as the number of fentanyls grow. So there was already a number of well-known analogs, such as 3-methylfentanyl, known to have the piperidine 1, 2, 3, 4 numbering system. So we kept that nomenclature. But there are the phenyl rings as well. So for substituents that are on the N-alkyl chain arrow ring, we have started using 2', 3', and 4', and so on around the ring. For the other anyl ring, which is part of the aniline ring system, we use ortho, meta, and para, or O, M, and P. And the ethyl bridge of the phenethylamine group was already well established and named alpha and beta, so we didn't make any changes to that. But the propionyl chain here, or the amide group, if not swapped out for an entirely different named group like a phenyl or a thiophene or crotonil, as previously discussed, posed a challenge. So we decided to start using alpha prime, beta prime, etc. for that area. So to clarify, I've drawn some examples of known fentanyls and left the guide in the upper right-hand corner. So the compound in the upper left, let me just move the arrow over here, that compound was recently described in a drug testing and analysis article, but the way it was named in that paper was ambiguous. Using our naming convention, this molecule would be called 2'-fluoro orthofluorofentanyl. If there is a methoxy off the first carbon past the amide carbonyl, that would be named alpha'-methoxyfentanyl. Substituents on the aniline ring are given the standard organic chemistry designations ortho, meta, and para, as you see in this example, and this one down here. Substituents on the piperidine ring, I mentioned 3-methylfentanyl earlier, that uses a normal 2, 3, 4 numbering system. To differentiate it from substituents on this arrow ring, so here's an example, 4'-methylfentanyl. And lastly, you see the alpha-methylfentanyl showing the substituents on the ethyl bridge and how those are named. So let me get to my next slide. Okay, so the key points that I would like you to remember from this section is that the emergence of novel synthetic opioids, particularly the fentanyls, is rapid, and there are no signs of this trend slowing down. Cayman Chemical is closely monitoring the market and is working toward providing the most needed analytical reference standards as quickly as possible. Cayman scientists have developed a standardized way of naming the new substituted fentanyls. And lastly, the point I want to make before I turn it over to Roxanne is please let us know what you need. Industry is your partner, and we are here to provide the tools and resources you need. So I will now turn it over to Roxanne Franskowski, our ISO Quality Manager.
Thanks, Donna. We just had a quick seating change there, so. All right, so I am actually going to piggyback on what Donna was saying about responding to the community. She was able to show you the thought process in R&D that goes into synthesizing the materials. However, once the materials are made, they need to be analyzed to ensure the correct material was synthesized. This is where QC and QA come into play. Most of the time our products do not have any issues and will pass QC testing. But there are the rare occasions we need to be the enforcer because the material didn't meet a specification, needs to be kicked back for further work. But this shows that the quality system works, and it is important that it does. As forensic labs were being accredited under ISO 17025, there was a need for high-quality, well-characterized reference materials. Under ISO accreditation, laboratories are required, whenever possible, to use reference materials that are metrologically traceable to the SI unit. In the forensic field, it is especially critical when there are lives literally dependent on the outcome of your analyses. Several laboratories were accredited under ASCLD Lab, which is now part of ANAB. This is why I'm going to cite ANAB document here. And for the record, I'm just going to throw out a disclaimer that there are other accrediting bodies, and I nor Cayman are explicitly endorsing ANAB over any other accrediting body. So to get to the ANAB document for forensic testing laboratories accreditation requirements, in section 5.6.3.2.1, that's a mouthful, if available, suppliers of certified reference material used to establish or maintain measurement traceability shall either be a National Metrology Institute, which in the U.S. that would be NIST, or B, an accredited reference material producer that is accredited to ISO Guide 34 with a scope of accreditation covering the certified reference material. So that's in actual accreditation documents. The next thing I'd like to point out is the SWIG drug recommendations. So if you're out there and you have not read the SWIG drug recommendations, I would recommend that you actually do that because it's well written, it's well thought out, there's a lot of great information in there. But the analytical procedures for drug analysis in Part 4A, Section 6.1.6, says reference materials and reference data are critical to demonstrating the validity of quantitative and qualitative test results. And further in the document, under 6.2.6.1, for reference materials obtained from a provider accredited under ISO Guide 34, the information contained in the accompanying certificate is considered reliable and can be accepted as correct if the material is stored and used in accordance with the manufacturer's instructions. So as you noticed, the previous slides, I highlighted ISO Guide 34. Many of you are aware that ISO 17025 is a standard for testing and calibration laboratories, but ISO Guide 34 is the guidance for reference material producers to show competency in the manufacture of reference materials. Some of you may remember a few years back when Cayman was first becoming noticed in the forensics field, and I think many of us in forensics were kind of questioning who Cayman was and are they a legit company. Well, first, yes, I think everyone has learned that Cayman is definitely legitimate, but one way to respond to the forensic community and meet the needs is that Cayman became accredited under ISO 17025 and ISO Guide 34 in 2013. You are able to find our ISO certificates and scope of analysis on our website, as well as an example of our C of A if you go to the forensic menu and select Quality and Certifications. I'd like to note that any accredited reference material provider will make their certificates and scope of accreditation available. If a RMP states they meet ISO requirements, that does not mean they are an ISO accredited laboratory. So buyer beware, I guess, is my point to make there. So updates, I just want to point out a couple updates that are happening in the ISO world. So in November of 2016, ISO 17034 was published, which actually replaces ISO Guide 34. So in essence, the reference material producers now have their own ISO standard to meet as opposed to a guidance document. The upgrade to standard includes requirements for production of all types of reference materials, additional specified requirements for CRMs. There's a harmonization with the revisions of ISO Guide 31, which dictates certificates, labels, and documentation, and Guide 35, which covers statistics, stability, homogeneity, uncertainty, and also is the inclusion of risks and opportunities into this standard. RMPs will actually have 3 years from the publication of ISO 17034 to be accredited under the standard. So in 2019, any Guide 34 facility seeking reaccreditation will need to do so under ISO 17034. As some of you may be aware, ISO 17025 is in the process of being updated and is currently in the final publication stages. The last I had heard, it is anticipated to be published at the end of this year or early next year. From what I've seen, one of the biggest changes to ISO 17025 is moving towards a more risk assessment-based system as opposed to a prescriptive-based system. This update lines up nicely with the 17034 publication. And like with the ISO 17034, ISO 17025, once it's published, will have a 3-year transition period. So I hope you all stayed awake through that. Now let's move on to our next topic. If we can pull up the question for this, I'd like to get your feel on reference materials and certified reference materials. So the question we have is, what is your primary purpose for using a reference material? And I already see a bunch of people answering. Yes. So the reason we're going to discuss this, what are they and what are the differences? All right, so I see over half of them use it for qualitative purposes and some of them use for both. So, okay, I think that's a really good representation. So, okay, I'm going to go ahead and move on now. So a reference material. A reference material is classified as being sufficiently homogeneous and stable. It's fit for its intended use in measurement or an examination of nominal properties. So what does fit for its intended use mean? Well, how do you plan on using the material? Is it for qualitative or quantitative purposes? Do you need an ultra-pure reference material, or can you attain the same results using a slightly pure material? If you have a mixture, do all the components need to be at 1 mg per mil because you're quantitating, or are you only trying to confirm retention time and or spectral identification? So would a 0.9 mg per mil fit that purpose? So it's up to the user to decide what the purpose of the material is going to be so the correct grade of material can actually be acquired. Reference materials may or may not have assigned quantity values, but positive structural confirmation of the material is required. And the traceability of the material. Where did this material come from? Are you able to trace it back to the origin of material? Does the material have unique identifiers such as item and batch numbers? Now, certified reference materials. Certified reference materials are accompanied by a certificate. Not all items with a certificate of analysis are a CRM. They may just be a reference material. Oh, lost my screen. Okay, here we go. So certified concentrations with uncertainty is what differentiates the CRMs from RMs. And the traceability referred to in the slide is the metrological traceability, measurement that's actually traceable back to the SI unit. And this is really important when you need to quantitate material. So how do we characterize our RMs and our CRMs? The next few slides, we're actually going to go through what that process looks like in our laboratory. So I'd like to bring us back to this scheme that Donna had shown us earlier in the presentation. And we're going to go through the process, but we're only going to concentrate on the QC of the compound. So once a material has been submitted for testing, each material undergoes the following QC process. And I've broken it down into 4 main components: method validation, RM characterization of the neat materials, CRM characterizations of the solutions, and stability studies. So for method validation, I've listed chromatographic method validation because that's the most common validation we perform. So it is done for each individual compound. And this can be done with HPLC or GC. I would say HPLC is definitely our heavy lifter in our laboratory. We're looking for things such as selectivity, sensitivity, linearity, precision, etc. So, but I would like to note, whenever a new technique is introduced, it is validated as well. For example, we introduced a GCMS chiral purity test for methamphetamine. This method was thoroughly validated prior to testing being performed. Next, I'd like to go over the RM characterization of neat materials. So this includes the purity and identity of the neat material. It's determined through a series of tests. We have our chromatographic purity. We have volatile determination, which includes loss on drying, Karl-Fischer water determination, and residual solvent analysis. It could be a combination of the tests or just one of the tests for volatile determination. It is really compound dependent. We do inorganic content is determined by residue on ignition. So chromatographic purity, volatile determination, and inorganic content are measurements that contribute to the estimate of uncertainty, which uncertainty is very big in the ISO world. Oh, sorry, I'm going to go back. Structural identification, cannot leave that out. This is determined using a combination of GCMS, LCMS, FTI, and NMR. I threw chiral determination up there, but it's only used on certain materials as needed. All right, now I'm going to move on to CRM characterization. So once the neat material is fully characterized, then that material is actually turned back over to our production to make the CRM solution. So that process, they actually take the neat material, weigh it out on ISO equipment that's been validated under our ISO system, and is ampuled and returned back to the QC department to complete that characterization process. And that includes concentration verification and homogeneity testing. And because there are so many, there's a lot of instrument time that goes into this process, because our homogeneity sometimes takes us well over a day of just instrument time. So with homogeneity, there's a within bottle and bottle-to-bottle homogeneity test that's performed. And then homogeneity also contributes to the estimate of uncertainty. So once all that data is collected, that needs to be reprocessed. So it doesn't look like a lot, but it actually is a very time-consuming process. Last, but certainly not least, on the important scale is our stability testing. Short-term stability testing is an accelerated study conducted over a period of several days. The materials are put into extreme conditions to determine the rate of degradation. Short-term stability studies are good for determining shipping conditions. The long-term stability studies are conducted in real time over a period of years, and the materials are kept at the recommended storage conditions. Stability testing is also something that is factored into the estimate of uncertainty. Back screen. Okay, here we go. Our screen keeps blanking out over here, so I apologize. This is our certificate of analysis. There's just a couple of things I'd like to point out to you. Once all of our data is collected, we have a great QA staff that actually puts these certificates of analysis together. So, but a couple of things I'd like to point out. There will be a measurement of uncertainty comments on the C of A. There you will have your certified concentration. So that's the actual concentration of the material with its established uncertainty, which should not be confused with the nominal concentration we have at the top here. We do the neat material characterization. So all of our testing we do on the neat material, those results are provided with the associated uncertainties as necessary. And then we also provide the instrumental data along with the conditions that we used on that for that testing. On this page, I'd like to point out the homogeneity testing results, and then we also have statements of stability for the materials. So what's really interesting, one of the new criteria under ISO 17034 is that C of A's must provide a recommended use as well, which is a reasonable estimate of stability based on how the customer is anticipated to use the material. So for example, a customer purchases a 1 mg per mil ampule. It is reasonable to provide a recommended as single use only. Conversely, the customer purchases 1 gram of neat material. It's unreasonable to assume that the customer will be using the material all at once. So a stability estimate for reasonable use will need to be determined. For example, this material can go through X amount of thaws and refreeze cycles before degradation was detected. This is a fairly new criteria, and it's something that we're in the process of tweaking out, but this will be something that we'll need to put on our certificates in the coming years. So now I'd like to move on and switch gears, and we're going to talk about other ways that we are responding to the community out there. What other services Cayman has been able to provide to the community? So here's a screenshot of our homepage, and if you go to the forensic page, you can, by the areas I have circled here in yellow, either in that top menu bar or that forensic science products button at the bottom, it'll get you to the forensic page, and you will see that we provide free web tools. So the first free web tool I'd like to talk about is our spectral library. It's free to download, and we try to update it quarterly with GCMS data. Our next update is scheduled for the end of November, and with the next update, we will be over 1,000 compounds. My group works really hard on putting this together, and we realize this is such an asset to the community that we're trying to be quick about it, but there's just a lot to add. So this library is designed to be used as a resource to help guide you. I would recommend confirming the identity of a material using your own mass spec instrument, though, and not you shouldn't completely rely on the library if you have a way to get the material. So if it's on our library, then you could probably purchase the material, and that's really how you should, as a forensic chemist, you probably should be confirming the identity of your structure. Now, another tool I'd like to point out is we have a GCMS search tool. Here you can search our database by, pull up here, by the formula weight. You can punch in the formula weight here and your base peak ion and any other, and the second most abundant ion you can put in there, and it'll generate a list of potential compounds that might match your spectrum. If you don't find a match for your spectrum in the GCMS library or with this search tool, please contact us, and we can most likely get it to you. We may have it, but the spectrum may not have made it to our website yet. So the next item I'd like to bring to attention is our most recent issue of Cayman Current. As we were beginning to see more and more fentanyls added to our GCMS library, my ISO QC manager asked if a paper on predictive fentanyl patterns would be useful to the forensic community. I thought this was a great idea, especially since forensic chemists and toxicologists are encountering more unknowns which have no spectral library match. Often they have to really look at the fragments in the GCMS to figure out what that tweak is on the molecule. So a group of scientists here at Cayman put together a GCMS interpretation guide, and it's available on our website. So in this publication, we provide the tools you need to help solve your unknowns. Excuse me. We also have a table of close to 100 fentanyls listed in order of descending base peak, something similar to what you would find on our GCMS tool. So the community's feedback is always welcome, and we are actually discussing ideas for our next current publication. So I'd like to kind of talk about the synthetic opioids and the overdose epidemic symposium that we hosted at the end of October. So Cayman often hosts symposia for other research areas, so we felt that the opioid epidemic is such a prevalent problem, and being partners with the forensic community, we believe that hosting a symposium for this topic would be beneficial. So we were fortunate to have Dr. Barry Logan and the Center of Forensic Science Research and Education, NIST, and Pinpoint Testing to host this event with us. It was a very busy 1.5 days, but the content was completely worth it. So approximately 150 stakeholders attended, and they spanned from forensics, toxicology, medical examiners, public health, law enforcement, academia, government agencies, private industry, domestic and foreign attendees were also present. So it was a very broad-ranging symposium. So I hope that those who attended found the content presented at the symposium to be useful. So there was a large amount of information shared at this event, but two of the big takeaways from the meeting were a need for enhanced analytical capability, for example, rapid reference material being provided, and analytical methods, being able to share analytical methods amongst the community. And also, collaborations amongst stakeholders need to be formed to develop an NPS surveillance system. So for those of you who are unable to attend the symposium, there will be a series of publications with recommendations coming out, and they'll show ways for stakeholders to become involved in the solutions and share information in real time. One of our symposium speakers, MJ Menendez, will actually be presenting on Wednesday, December 13th as a part of this webinar series. She was a phenomenal speaker. I recommend that our audience join that discussion and hear what she has to say. So last but certainly not least, safety. We often get asked for safety recommendations. It's not our area of expertise to offer that advice to laboratories, so my disclaimer for this section is that any information I'm about to present is informational only and should not be taken as any recommendation on how individual laboratories should establish their safety protocol or policies. So a question we usually see is, how much PPE should I use? And we hear this quite often from our customers. So I don't know that we actually need to use as much PPE as our 1918 friend here, but the CDC, the DEA, they actually put out some pretty good guidelines. There are other places that have put out guidelines as well. So I would actually refer you to those to help determine what and how much PPE you should actually use. But I would like to discuss, just kind of show you the different levels of PPE. So level A is an encapsulated suit. The user has a self-contained breathing apparatus, and they're typically specially trained to use this suit. Level B is a Tyvek resistant suit, or chemical resistant suit, or Tyvek. Again, they're using the SCBA, so that includes some special training, and gloves and boots to make sure all your digits are covered. So level C, we had our Tyvek suit, and you could use your air purifying respirator, gloves, and booties. Now this was actually during our carfentanil synthesis. Our safety team spent several hours coming up with the correct protocol for our facility when we were making carfentanil. And, so we decided that this was the appropriate level of protection we needed for all of those in the production of this material. Now, on the analytical level, during carfentanil, we decided we didn't need as much PPE because we weren't directly working with the meat material, but we did use disposable lab coats, the N95 masks, safety glasses, nitrile gloves, and shoe covers, just to protect us from any risk. again, the decision was based on the types of analysis we were doing and how exposed we would be to the neat material and solutions. Here we have our synthetic chemist on a standard day working in the lab. That center picture is Chung, our driver of the carfentanil synthesis. Hi, Chung. You notice that some people prefer the N95 mask, where others prefer the half-face respirator. It's all a preference on what you feel most comfortable with and what is appropriate for the situation. So site safety, we made sure to restrict access to only essential staff as we're going through our carfentanil production, as well as minimize risk for potential exposure. So here we have, we blocked off where we were doing the synthesis. The doors in our production lab were taped off. This was not our only exit. This is not our only exit door. There was another way out, so nobody had to run through caution tape in case of an emergency. Here, we did have a tent in place to kind of contain the material in case something fell. But these are not the only items that you could use to protect yourself. I read or saw a report the other day where GBI actually has powder hoods. So that's something that that's always an option as well. Again, site safety and personal safety needs to be a conversation between lab staff and safety personnel to determine the appropriate protocols for your facility. We are always communicating with our safety staff. We have some really awesome safety staff. Kim, Andrew, Mike. When we were manufacturing our carfentanil, our safety team was with us the whole way with the emergency kit close by. And we now have naloxone kits in the laboratories where chemists are handling opioids. And we have people trained on how to use them. So I guess for my part of the presentation, the takeaways I'd like to kind of leave you with are high-quality analytical reference standards take time to synthesize in QC. We wish we could get them out to you in a week, but sometimes it just isn't possible, but we are doing our best. The Cayman Web tools are available to assist you with your unknowns. Laboratory safety protocols should be handled at the agency level, but personal safety should be handled by the individual. And Cayman chemists are always here to assist you. As Donna said, we are here to partner with you. You'll have our contact information, so please reach out to us if you have any extra questions. And with that, I'd like to thank you. And now I would also like to acknowledge Josh and NFTCOE for inviting us again to be a part of this webinar series. The forensic community, it's a pleasure working with you guys. We love going to conferences and talking to everybody. Cayman Chemical and our forensic chemistry division, which is in the picture here, they are the hardest working, most dedicated group. Both Donna and I are honored to be able to work with such a great group of people every single day. So I guess with that, we can open it up to questions.
All right, thank you so much, Donna and Roxanne, for your presentation. We do have a few that have come in, so we'll just go ahead and jump right into it. Before we do ask the first question, again, Donna and Roxanne both wanted to make sure that you had their contact information. So at the very bottom of your screen there, you will see the email addresses for both of our presenters today. So definitely make sure that you take that down before you do leave the room. So our first question is from Christiana. She's asking, she said, thank you for the informative presentation. Do you happen to know about the prevalence of cyclopropylfentanyl on the street market?
It is, from what I can tell, it's quite widespread. It is often encountered in mixtures as well as counterfeit tablets. There are a few other fentanyls, such as allylfentanyl, crotonylfentanyl, fentanyl, and methacrylfentanyl, that have the same molecular weight and base peak. So it's quite possible that not every cyclopropylfentanyl case is cyclopropylfentanyl. But we've just begun discussing writing a white paper to share the comparative data that we have on all those fentanyls. So I would advise anyone to get on our email distribution list so that as soon as we have that publication handy, we can get it into your hands.
All right. Next question we have is from Chris Johnston. He's asking what up-and-coming drugs are being requested for future production?
We're getting requests on a variety of things. We're kind of surprised to be getting some requests for synthetic cannabinoids, as well as some cathinones and amphetamines. So that's why I mentioned earlier we can't lose sight of that. We're still seeing designer cathinones and amphetamines and tryptamines popping up. We're still getting requests for fentanyl analogs. We're getting data from some customers that seem to suggest some new U47700 analogs. So yeah, like I said, the list of fentanyl analogs that can be made is extremely long, and I don't really see an end in sight.
All right. Next question we have is from Dr. Leo. He's asking kind of on your opinion, why is fentanyl named ending in a YL while carfentanil ends in an IL? Were there any rules based beyond that decision?
That naming system wasn't developed by anyone here at Cayman. I think some of the reason has to do with the British spelling. Thank you. The British spelling of carfentanil versus the North American spelling of fentanyl itself. So I think if carfentanil, for example, may have been first discovered or developed in Europe, then maybe they started using that IL designation, and it just persisted when it moved, to North America. That's probably the best answer that I can give about that. Do you have any idea, Roxanne?
No. It's probably just what they patented it at, honestly.
Yeah, if you notice, a lot of the Schedule II drugs like sufentanil and thiofentanil and carfentanil, alfentanil have the I in the name, but most of the new designer drugs seem to have the YL. I've noticed that too, but I'm not sure I have a really good answer for you.
All right, so we have a few comments that came in, so I'll read the comments, and if you want to kind of elaborate on anything, please feel free to. So the first comment was from Steve Hokinson, and he said there was an email that was sent out today on the CLIC server about the naming ambiguity, how the synonym desmethylfentanyl is used for both acetylfentanyl and benzylfentanyl. He wanted to thank you for explaining the naming system that Cayman is using. It definitely helped him out.
Well, that's good to hear. We definitely recognized that there was a need for some consistency. So we're happy that we were able to provide that service.
All right, next comment came from Lisa Windsor. Our lab sent in our GCMS data to Cayman recently because we had an unidentified compound. Cayman helped us determine that our unknown is cyclopropylfentanyl. We've had 3 additional cases of tablets containing cyclopropylfentanyl since then. They wanted to say many thanks to Cayman for helping them out with their issues.
You are quite welcome.
Yeah. No. It's glad. I'm glad we're able to help in that respect. So. Yeah, any way we can help out, we're always willing to, kind of help you guys out, and if we can provide data to you or whatever, we're, like we said, we're here to help. So I'm glad to see we're getting some positive feedback on those efforts.
So we did have a question come in from Brett Brooks. He's asking, from a law enforcement perspective, would normal CPR masks protect the user from possible cross-contamination?
That's kind of out of the realm of what we do, so I don't know that I would actually have a good answer for you on that, unfortunately.
Yeah, I don't have an answer either. I don't even know what a CPR mask is, actually, so
yeah.
That's not our area of expertise.
Yeah, sorry, I'm not able to answer that for you.
Not a problem at all. We have a few people typing, but we'll move on to another comment that came on from Christiana saying, thanks, Lisa, for the feedback from another user. It seems that cyclopropylfentanyl is still another addition to the street market. They have recently identified it by high-resolution accurate mass, but there is nothing published about it just yet. All right, next question we have that just came in from Kristen. Do you provide reference standards for CIS trans 3-methylfentanyl separately? And do you know which is more requested by your clients?
Yes, we do offer the isolated cis and 3-methylfentanyl compounds. I do know that the cis 3-methylfentanyl is the more active of the two isomers, and I believe in the mixtures that are trafficked on the street, it is the more prevalent of the two.
All right, that was our last question of the day. So we're going to go ahead and move into our closing, but before we do, I would definitely like to ask our speakers if there's anything that they would like to close us out, any additional knowledge they would like to give out to our audience today.
Thanks, Josh. Yeah, I mean, if you can think of any additional questions that you have for how Cayman can help, please don't hesitate to contact our technical support team, our sales team, myself, or Roxanne. Thank you, Josh, and the Forensic Technology Center of Excellence for this invitation to participate in this webinar series. And thank you to the audience today for your comments, for your attention, and for the very good questions. And enjoy your Thanksgiving holiday next week.
Yep, and Donna pretty much wrapped it up for me, so I just want to mirror her comments. And yeah, happy Thanksgiving, everyone.
Thank you so much. Obviously, we would like to thank our subject matter experts and presenters today, Donna Ayula and Roxanne Frankowski, for their presentation. We definitely appreciate them taking the time out of their day to present the material for today's webinar. We would also like to thank our audience members for attending and joining us today. These webinars are definitely not possible without your attendance and your support. As always, before you leave today's webinar, we do ask that you participate in our short survey. You will find it there in the middle of your screen. Simply click the word "Here" and a new window will pop up for you. These surveys are vital information that we always use to help provide the best webinar experience, as well as the content that we disseminate out to our audience. So definitely, if you wouldn't mind, please take a few moments to complete the survey. We would definitely appreciate it. If you look in the bottom left-hand corner of your screen, you will see a PDF version of today's slide deck. So before you do leave today, go ahead and click on the title of that file. The Download Files button will be highlighted for you at the bottom of that pod, and you can download their slides directly from the room today. So please make sure you do that before you leave so that you have a copy. Also, popping up on your screen now is a new window with our website. So please go check out all of our live upcoming webinars as well as all of our archival webinars. We are recording this webinar as we do with all of our other webinars. And then we do build an archival version of those webinars so that you can reference them back or provide them to any colleagues that might have missed today's webinar. This recording should be up in the next few days. So definitely make sure to check it out if you would like to reference back. Again, thank you, Donna and Roxanne, for your presentation. Thank you, all of our audience members, for joining us today, and we hope everyone has a great